Today we are looking at the glitches in the human blueprint. Genetic disorders. Genetic disorders aren't just one thing. They actually fall into two very different categories based on how big the mistake is. The first one is Mendelian, and the second one is chromosomal. Think of a DNA as a library. A Mendelian disorder is like a tiny typo on one single page. That means mistake or mutation in one single gene. While the chromosomal disorder is like having an entire extra book in the library or a whole book is missing. So here the mutation will be on the whole chromosome. Let's deep dive into Mendelian genetic disorders. Single gene will be mutated over here. These are caused by an alteration in just one single gene. Because it's a gene level issue, it follows the laws of inheritance that Gregor Mendel discovered. It follows the laws of inheritance discovered by Mendel. Now, to understand the rules, you have to ask two questions to yourself. First is, where is it? Where? Where is the mutation is? Is it on your autosomal chromosome or sex-linked chromosome? If the mutation is on first to 22nd pair of chromosome, it will called autosomal disorder. And if it is on 23rd, the last pair on X or Y chromosome, then it is sex-linked. And the second question is, how strong? Is based on if the mutation is on dominant gene or on recessive gene. If the mutation is on dominant gene, for example, A is dominant and A is recessive. So there will be three possibilities. If this is causing the disorder, only one gene will be enough to get the disorder. And what if the problem is on the recessive gene? Again, three possibilities. The disorder will be only on this particular pair. but this will act as carrier. This person will not have any active symptoms, but it will act as carrier. What does that mean? If this person mates with the similar genes, there are four possibilities. First is capital A, capital A, capital A, small a, again capital A, small a, and small a, small a. So he or she will be having 25% of possibility to get the disorder. If the person is carrier, we will go into detail afterwards. Now let's deep dive into chromosomal disorders. So, as we saw, chromosomal disorders happen when the entire chromosome is affected. Not a single gene. The entire chromosome is affected. It's a quantity error. How and why this happens? This happens simply because of non-disjunction. What does that mean? It simply means that during the cell division, the chromosomes got stuck and didn't separate properly. One cell gets too many and one gets too few. For example, if this is a cell and during cell division, instead of separating from here, they separate differently from here maybe. So the one cell will get more of the DNA and the other cell will be left with very few. This is non-disjunction. For example, in aneuploidy, what happens? In this you have one extra chromosome. One extra chromosome. For example, Down syndrome. In Down syndrome, you have trisomy, which is having 3 chromosomes in a pair instead of 2. Now, what happens if you have only 1 chromosome instead of 2? That is called monosomy. Mono means 1. In monosomy, you will only have 1 chromosome. The entire 1 chromosome is missing. So it's all about the balance of DNA. So there you have it. Mendelian is a single gene typo and chromosomal is a whole book error. In our next video, we'll look at the specific diseases like sickle cell and Down syndrome in detail. Make sure to subscribe so you don't miss it. Thank you for watching.